Hello, everyone. Welcome to the Medican Express webinar series. My name is Ian Wan. It is our pleasure to have with us today Dr. Michel Poussiat, Senior Research Directors and Group Leaders at the French National Institute of Health and Medical Research, also known as INSERM. Dr. Poussiat currently leads groundbreaking research in physiopathologies of cardiac development at Faculty of Medicine in Marseille, France. Dr. Poussiat's extensive research career spans over 3 decades. He made his significant contributions to the fields of basic cardiac physiology and cardiology, with a particular focus on various diseases associated with cardiac development, such as myocardial and valvular development, malfunction formations, and for today's topic, Cornelia de Lange Syndrome, or CdLS. His team uses a number of innovative approaches, such as novel mouse models, cardiac organoids, and advanced multi-omics and bioinformatics techniques. He has an impressive list of publications with 139 research papers and over 7,200 citations on his credits. Among his works are studies on cell reprogramming in cardiac valve developments and the role of histone demethylase in preventing cardiomyopathy. Currently, Dr. Poussiat is actively involved in multiple high-impact grants, including research on cardiac organoids, which aims to revolutionize our approach to cardiac developments and drug discovery. So please join me in welcoming Dr. Michel Poussiat, who today will share his insights on latest advancements in cellular senescence in Cornelia de Lange syndrome and possible therapeutic approaches to combat this disease. Let's give a warm welcome to Dr. Poussiat.
Yep. Thank you very much for this nice introduction.
Yep.
So I'm going to share my screen now. Okay. Is it fine?
Yeah.
Okay. So let's start. Indeed, so today I'm going to talk about a rare syndrome, Cornelia de Lange syndrome, that is called indeed as abbreviated CdLS. So first, just to tell you about this syndrome, because this is really a tremendous and a very deleterious syndrome. So this is a rare syndrome. It's about 1 to 10,000 births, newborn. But in fact, there is now, we know that there is about 8,000 patients in Europe, about the same number in US. So basically, it's a delay, in fact, in development. So there is a slow growth before and after birth. And this is associated with a lot of different defects in many different organs. So the first impacted organ is, in fact, the brain, with a lot of intellectual disabilities, autistic syndrome, even self-destructive behaviors. There are some skeletal abnormalities because there is indeed there is a problem in the development of the skeleton. That's why people you will see are short. There are some gastrointestinal problems, hearing loss in some of the kids, myopia, and congenital heart defect. That's why we came to this syndrome, because you will understand this is in fact, of course, a genetic syndrome, which is associated with a lot of epigenetic problems. So there are several genes which have been involved in this syndrome, but the main one, which is responsible for about 8% of this syndrome, is NIPBL. I will show you what is this gene and what is this function right after this slide. But first, what you have to know that this is a de novo mutations. So this is what is very bad surprise for the parents, because it's really de novo. And most of the kids, even now, they are more and more detected during fetal life. In fact, most of these patients were detected or were only at birth. And there is a mosaicism, so which explain that not the patients may have a very mild or very severe syndrome. It just depends when during development the mutation did occur. So what is NIPBL? So NIPBL, I don't know if you see the mouse. Yeah, okay. Yeah. So NIPBL is one of the components of what we call the coenzyme complex. The coenzyme complex was known for many, many years, as you know, because this is a complex which is responsible for the cohesion of the sister chromatid. But what we learned already more than 10 years ago is that this coenzyme complex is very important for gene transcriptions because it makes a loop between two DNA, two different regions of the DNA. And for example, it makes a loop. And this is a way that an enhancer, which could be quite far away from the promoters, is folded on the promoters, and the coenzyme complex can maintain this interaction, DNA-DNA interactions. So you can imagine that this coenzyme complex during transcription is loaded and unloaded when it is required and then unrequired. And there are two proteins that load the complex onto DNA, and these proteins are indeed NAPBL and MAO2. So we don't know too many mutations in MAO2. We know most of the mutations responsible again for CdLS on NIPBL. So this is a key genes because this is of course a genes when it is mutated that is responsible for a lot of defect in transcription, gene transcriptions. So the patients have in fact haploinsufficiency in these genes. This is a very mild haploinsufficiency because you can understand that too much haploinsufficiency and then the cells cannot divide anymore. And of course, there is no individual which can survive. So we basically assume that there is about 20% haploinsufficiency in these genes. So I have to introduce right now another concept, which is not related directly to this syndrome, but that I will use and I will explain why we came to this program cell senescence. So the program cell senescence during mammalian embryonic development, in fact, is a quite new concept. It has been, as you can see, published just about 10 years ago by two Spanish labs in Cell paper, back-to-back Cell paper, where these people identify that indeed there is cell senescence during embryonic development. It's quite surprising at the first approach because the organism is developed, there is a lot of division, cell division, and so on. So what about cell senescence? So cell senescence is in fact cell replicative senescence. Okay, so it has nothing to do with aging. It is really the first step toward aging, but during development, in fact, cell senescence is basically a mechanism which is there to eliminate the structure that we don't need anymore when we get adults. So this is a very amazing phenomenon that the nature could basically turn on. And these people have shown that indeed there are some organs that undergo cell senescence, not organ, but tissue, sorry, during development. So we use as marker of cell senescence mostly the histone, the gamma H2AX histone, which is very well known to be induced in fact in stress-induced senescence, and P21, which is a CDK inhibitors. So indeed, we decided to generate, in fact, a mouse to study this CdLS because there was already a mouse generated by an American lab, which was a mouse in another genetic background, which recapitulated very, very sparsely, I would say, the syndrome, only 20% and not all the phenotype of the patients. So we decided to backcross this mouse, which is a NAPBL plus minus. So this is an haploinsufficient mouse for NAPBL genes in the C57 Black Cis genetic background. And you see on this slide first that already during embryogenesis at 13.5 in the mouse, you see that the NAPBL plus minus embryo are much smaller. they are still very much smaller at birth. This is P1, the next one. And then you see even the mice, when they're adult at 1 month, clearly these mice are smaller than the wild type. So exactly like the patients, which are much shorter than the normal. So we look more carefully at the skeleton. And indeed, when we measure, for example, the tibia, we measure the rib thickness, we measure the finger length here. And you see that compared to the wild type, most of these bones are indeed shorter in the mice. Both the tibia length, the thickness of this rib, you can see from wild type and NEPBL plus minus, and the finger. So then we look in detail into this bone, and what we discover that in fact there was a decrease in the length of both the proliferating and the hypertrophic chondrocyte. You see again on the left the wild type, the plus plus, and the plus minus haploinsufficient NEPBL mouse. And you see that this zone, which is a proliferative zone, here is much shorter in, for example, in the NEPBL plus minus. You see here the difference, clear difference, again, in the length of the radius, and you see the hypertrophic zone here, which is decreased in the NEPBL plus minus. We did some staining as well, of collagens, which are collagen highly expressed in bone, and you see that both collagen 2 from collagen 10 is clearly decreased in the NEPBL plus minus mice. So now, what about the cardiac defect? So looking first in neonatal mice, we did some episcopy, high-resolution episcopy microscopy to look at the different malformation that these neonatal mice could basically feature. You see again on the top here, the wild type, and at the bottom panels, the NEPBL plus minus mice. So you can easily see at first glance that there is a huge hypertrophy. You see here the wall of the left ventricle. You see this huge, very thick wall of the left ventricle. But very interestingly, there is as well very clear malformations, because if you look at the wild type, as expected, you have the outflow tract from the left ventricle, so the aorta, and the outflow tract from the right ventricle, the pulmonary trunk. But when you see at the NEPBL plus minus mice, you see that there is only one outlet, one outflow tract. There is no septation between the aortic and the pulmonary trunk. We look at the valve, and you see here, for example, the aortic valve, which is enlarged. This is better seen with the different staining here. You see very smaller leaflet here and much thicker leaflet. This is exactly the same magnification on both panels. And this is just the quantification of the thickness of the ventricular wall. Again, we look at more carefully at this heart. We detected a clear asymmetry between the right and the left ventricle. For example, which is very typical. The right ventricle has this crescent shape. You see here the chamber, the right ventricular chamber. Here you see the left ventricular chamber. And you see in the plus or minus mice that there is first a septal defect here. You see that the septum, which is supposed to separate both chambers, is not closed. There is a communications, which is very frequent in CdLS kids. And you see again that there is the wall of the right ventricle has not these crescent shapes, it's quite straight. And you see a difference because you see that there is almost the same volume in both chambers compared to the wild type. And we even detected, so this is detected in 90% of the mice. And in some mice, only in some mice, we even detected some interruption of the aortic arch that you can see here. So clearly, this mouse can recapitulate many cardiac malformations that are observed in the patients. So then we wanted to look at how this malformation during development can translate into aortic problems at the adult stage. So we look at the adult mice, And we did echocardiography. We did more specifically Doppler echocardiography to look at the aortic flux in these mice. So you see again the wild type, and you see the aortic flux here in NEPBL plus or minus. And you see that this flux is clearly increased in this mice. You see again here the first you see the velocity. Okay, so this is the velocity. This is what you see here, which is quantified here. So there is an increase again in the aortic flow. We measure the aortic diameter, and we see that there was indeed a decrease in the diameter of the aortic. So there is an aortic stenosis, which explains that the flow is increased. And because of this aortic, likely because of this aortic stenosis, the heart, of course, has more demand in terms of contractility and start to hypertrophy. So that's why we could see this hypertrophy and this increase ventricular wall, which is typical of the response of the heart to an aortic stenosis. So then, when we look at a little bit of the phenotype of this aorta, you see here the wild type aorta, which is stained by smooth muscle actin in green here, and you see this thick aortic wall here in the NEPBL plus or minus mice. But more than the thickness of the aortic wall, what was very, very striking is that you see that this aortic wall is full of smooth muscle cells, what is expected. This is the media of the aorta. But you see that in NEPBL plus or minus, you don't see any more SMA staining. And it looks that these cells in the media of the aorta lost their phenotype. And when we stain with a gamma H2AX, we found, in fact, that these cells indeed were gamma H2AX positive. So they look to be at least some feature of senescence. So already we could see that there is a problem here in the differentiation or maybe in the proliferation of smooth muscle cells in the aorta. So we went back to embryonic stage, and we look indeed in this embryonic heart. You see here the heart at E13.5 of development. You see again what I showed you before, that in wild type, you see clearly the two outlet, pulmonary artery and the aortic artery here. And we see that you see in NEPBL plus or minus, but there is only one outlet here, which basically communicate with the two chambers. And then we decided to stain with P21 to see if the problem of septation between the pulmonary and aortic trunk was indeed due to cell senescence, because we know that these septations, in fact, occur because of the migration of smooth muscle cells from, in fact, the basis of this aortic trunk. And indeed, when you see in green P21, you see a few cells. It's better seen here in the panel where you have only two staining, P21 and phospho arc in red. But if you see first, and if you focus in P21, you see only a few green nuclei compared to the NEPBL plus or minus, or you can see a lot of nuclei which are positive for P21. Here you see the quantification. We have many P21 positive cells at the basis of the aortic, which is a first index that indeed the smooth muscle cell, which was supposed to migrate to make the septation between the aortic and the pulmonary flow, could not probably do it because they were, in fact, becoming senescence. And we even detected a phospho arc increase in this aortic region. So to learn much better about what was going on, we decided to do both a RNA-seq approach and a proteomic approach. So we dissected out this outflow tract, embryonic outflow tract, and we did indeed RNA-seq, and you can see here a lot of genes which are expressed, overexpressed, or underexpressed. And what among these genes, we were very struck by the fact that TGF-β2, which we know is very important for septation of the outflow tract, and which is as well important for cell senescence. TGF-β pathway is a very well-known of cell senescence pathway. TGF-β or beta-2 specifically was really overexpressed. We did some qPCR to validate, and indeed there is much more TGF-β2 gene expression in the NEPBL plus minus outflow tract during embryogenesis already. And when we look at the proteomics, we detected a lot of proteins which are very important for the development of this outflow tract and pulmonary and the aortic trunk, which are also proteins which are known to be, again, in fact, mutated in different syndrome, which are aortic syndrome, amylin, fibrillin, fibronectin, and so on, which are indeed clearly downregulated in the NEPBL plus or minus max. So we decided to look in other regions of the embryo to look whether cell senescence was indeed a problem in the CdLS syndrome. Here, as I showed you at the beginning, I showed you that the bone were shorter, but there was a problem in the hypertrophic chondrocyte proliferations. And so we decided to look at early in the embryogenesis, again at E13.5, at the four-link cartilage primordium. And you see this primordium, which is stained by all these chondrocytes are stained by MSX1 in red. And again, P21 here is in white. So you see the wild type, you see a few, in fact, patches of P21 positive nuclei. But when you look at NEPBL plus or minus embryos, you see that most of this cartilage is positive for P21. So in fact, the cartilage plate very early on is already getting senescence, which explains why afterwards the bone cannot grow to the normal size. So then I have to introduce with a knowledge that people who are not working on cardiac development, of course, cannot know very well. In fact, heart developed from two different embryonic fields, what we call embryonic fields, the first and second embryonic fields. To summarize, the first embryonic fields give rise to our left ventricle, and the second one to the rest of the heart. But which is very, as well, amazing is that the second, in fact, heart field give also rise to a lot of muscle, which are, in fact, muscle of the face and muscle of the upper part of the body, of the neck, and even part of the muscle of the esophagus. So this is the same embryonic origin as the cardiac origins. So I told you that, in fact, these kids have problems as well with regurgitations, with esophageal and stomach regurgitations. So we were wondering whether it's not due to a defect in this esophageal muscle. And so we decided again to look in the embryos. So here we deleted NEPBL specifically in the second heart field with this mouse, with a MEF2C Cree mouse. So you see the heterozygous without any phenotype and the homozygous, where NEPBL is fully deleted, again, specifically in the second heart field. And I'm not going to go into detail because there are many, many muscles here in this slide. But if you look, for example, at the esophagus muscle that you can see clearly here, or you see here, and when you look at the NEPBL flux mice, you almost barely see this muscle. You see very, very low developed muscle. This is the same for the trapezius muscle, which is one of our muscles of the neck. You see the very nice developed trapezius muscle. And here you see that this muscle is almost missing. And there are a lot, you see as well the masseter here. There are a lot of muscle what we know are derived from the second heart field, which are indeed less developed, underdeveloped in these mice when we delete NEPBL specifically in these cardiac territories. So looking in adult now at this esophagus skeletal muscle, I'm talking here about the skeletal muscle of the esophagus, because when you think about the esophagus, we are always thinking about the smooth muscle. But there is, besides the smooth muscle, that you can see here in red, there is as well a skeletal muscle which is surrounding the skeletal muscle, which is very important for the contraction of this muscle. And again, we stain for P21, and you see the wild type. And you see here clearly a lot of P21 positive cells in the skeletal muscle. This is in this part, actinin positive part. And we measure the thickness of the skeletal muscle. And you see about 50 micrometers thickness in the wild type compared to 18 micrometers in NEPBL plus or minus. So clearly what we could see before, that this muscle was not very well developed, this corresponds again at the adult stage of a very thin skeletal muscle of this esophagus, and this is likely due again to cell senescence. So then I told you that the most and what is in fact impacting at 100% these kids are indeed brain defects, any kind. A collaborator years ago had published a nice paper in Neuron showing that there is a problem of cortical neuron migrations. And we thought about this problem of migration, like I told you about migration of smooth muscle cell, which makes the septation of the outflow tract. So we look at the cortex, at the brain cortex of this mouse. These are neonatal brain. And this is basically the cortex area. And again, when you look at P21 positive cells, you see a lot of P21 positive cells in this cortical brain compared to the wild type. Okay, so what could be indeed this dysregulated, all this dysregulated molecular mechanism in NEPBL plus or minus? And we wanted to look whether what we found in mice, and specifically the cell senescence phenotype that we could see in different territories in many different regions of this embryonic and adult mice, were indeed something which could be reminiscent to the CdLS, so in patients. So we decided to derive iPS cells from different children's affected by this syndrome, and specifically children which had indeed featured some cardiac malformations, including aortic and pulmonary stenosis. So here you see this Cornelia de Lange patient-specific iPS cells, and we were very surprised because it has never reported in any iPS cell line that has been derived in the literature, is that there is already, when the cells are undifferentiated, some cell senescence in this iPS. And how we could figure that out is that when you look at the nuclei, the iPS, like any pluripotent stem cell, have two nuclei. Two nucleoli, one nucleus, of course, but two nucleoli in per nucleus. But here you don't see two, you see 3, 4, 5. Indeed, this is due to a problem of a breaking of this nucleoli. So we even quantify this, and we could see clearly that most of these nucleoli were broken in the NEPBL plus or minus iPS cells. And when you stain with for NIPBL, you see clearly a very nice NEPBL staining, quite homogeneous in the nucleus of healthy people. And you see in these patients that there are just patches of NEPBL. And more interestingly, it is NEPBL seems to be excluded from this nuclei, and specifically the broken nuclei. So as I told you a lot about smooth muscle cells, and we know that in different aging syndrome, smooth muscle cells are really the most impacted cell type by cell senescence. So we decided to derive and to differentiate the iPS cells specifically in smooth muscle cells. So thanks to the growth factor and small molecule that we are getting from MCE. So basically, it's a very simple protocol. So we know how to derive, of course, mesodermal cells with Wnt pathway, BMP, and then Wnt inhibitors. And then from the mesodermal cells that we can sort out, we just to add PDGF alpha to get smooth muscle cells. Again, you see in the white type here, the smooth muscle actin in green with the nuclei blue, and P21. So you don't see any P21, or if you look at the field, the whole field, you see one P21 positive cells in the wild type. But when you look at the smooth muscle cell of CdLS patients, you see that the cells don't look very healthy. They don't have this spread, in fact, shape. And you see again a lot of P21 positive nuclei. And you see basically here that most of the cells become senescence when they get differentiated into smooth muscle cells. So this cell senescence pathway during embryogenesis seems to occur as well in cell from patients, at least in smooth muscle cells. So now we have two, I would say, keywords. We have TGF-β, which is overexpressed, and we know that this is very important for cell senescence and cell senescence. So we have some ideas how to maybe target this syndrome. And then we ask ourselves, could we use an inhibitor of TGF-β pathway to at least alleviate some CdLS symptoms? So there is indeed several TGF-β receptor inhibitors, and there is specifically one which is under clinical trial to block specifically metastasis in different cancer. So we use these gallium-inserted molecules first to treat these iPS cells. Let's try in vitro and again in human cells. So you see again these P21 positive cells in CdLS, iPS cells derived into smooth muscle cell. And you see that when these cells are treated with the TGF-β inhibitors, with gallium-inserted, you don't see anymore. And you see that the cells start to spread and they look much more normal than they were looking before. Then we decided to treat the mice. So we decided to treat the pregnant mice and then to look at the embryos. And again, we look at the septation of the outflow tract. You see that these treated mice, treated wild type as expected as the two outlets, again, the aortic outlet, aortic trunk, and the pulmonary trunk. And you see again here that in the NEPBL plus or minus, now treated, you find back, in fact, the pulmonary tract getting into the right ventricle and the aortic trunk here getting into, as expected, the left ventricle. So it looks like that there is indeed, again, septations, and we could not detect anymore too many P21 positive cells in this aortic and pulmonary trunk. So we look then at birth. So you see again in untreated this cardiac hypertrophy, this very thick ventricular wall, left ventricular walls more specifically. And here you can see that in the treated mice, the neonates now get born with a normal heart, with a normal left ventricular wall. So there is no more difference between wild type and NAPBL plus or minus neonates. Then we look back at of course, at the bone, and again, we could not detect any more this delay in bone growth. Could see that there are the same, in fact, lengths in the hypertrophic chondrocyte, which of course explain the same lengths in the radius. So there is again no delay anymore in the bone growth. And indeed, we could not detect anymore any difference in COL2, in collagen 2 or collagen 10 staining into this bone of NEPBL plus or minus mice when the mother has been treated by these TGF-β receptors inhibitors. So again, this is just the whole skeleton. So same thing, no more difference. The mice look exactly the same size. The thickness of the rib, the tibial length, the finger length, everything was back to normal. So then we decided to look at the esophageal muscle. Again, you see very scarce P21 cells now, and you see a normal esophagus muscle, skeletal muscle. And then when we look at the brain cortex, again, there is very few P21. You see here the quantification, the wild type, many P21 positive cell in any PBL plus minus, but if the mother has been treated, the neonate don't show any difference in cell senescence in the brain context. So basically, this is the take-home message. In fact, what this syndrome looks like to be is in fact an exacerbation of this programmed embryonic cell senescence. In fact, I told you at the beginning, normally this is a normal process, which is very important to get rid out of structure, tissue that we don't need anymore during embryogenesis, but we don't need anymore when we get born. But in CdLS, it looks like that this process is absolutely dysregulated. So this process is exacerbated, probably because of TGF-β. So it means that more cells than expected are going to stop their proliferations. They are not going to migrate, and it's going to impact the morphogenesis of many organs. And this explains as well, it could explain why, in fact, the CdLS patients develop as well a quite accelerated aging during the adulthood, because indeed these patients develop aging-related disease when they are about 40 years old, which is quite young. So they develop osteoporosis, atherosclerosis, and a lot of diabetes, a lot of, we know, pathologies associated with aging. So this is a take-home message. This is going to be my final slide. Basically, what's going on in this NEPBL haploinsufficiency organism is that there is an exacerbation of the TGF-β pathway, which induces an exacerbation of embryonic cell senescence. And this leads to a lot of delay or defect in the formation of many tissues and organs, and leads finally to this neuronal defect, esophageal defect, cardiac defects, and a bone growth delay. So, but if we rescue by blocking this TGF-β pathway, by basically here in the mother, we can basically prevent most of these deleterious effects due to this exacerbating embryonic cell senescence. So then what we are going next, of course, it's too late to treat the kids at birth about cardiac defects, but it's not too late probably to treat these kids to try to decrease the impact of this syndrome on the development of the brain. As you know, the brain develops quite a lot after birth. And so we think that at least for the neuronal defect, there is a therapeutic windows at birth when the kids are detected as being a CdLS. And then I will thank also people, my student, Céline Faten, who work on this program, collaborator, a very helpful Erwan Vatrin in Rennes, who did a lot of, in fact, also proteomics and Western blot I didn't show you to characterize the mice. Valérie Cormier, the clinicians, Valérie Cormier there at Necker Hospital, who is in charge of the cohort, the French cohort of CdLS. Juan Pied, who is in charge of a huge cohorts and a very interesting and helpful clinics in Zaragoza in Spain of CdLS patients. Manuela Welling in Germany, who did all the work on bone. She's a specialist of bone development, and some other collaborators who gave us some insight into this work. And I thank you very much for your attention.
Yeah. Thanks, Dr. Pucet. It's a great talk. It's fascinating to see such a thorough analysis on the physiologies of a haploid insufficient mutation on single gene, and how you can use a pharmacological approach to reverse some of the phenotype. Let's see if the audience has any questions. Okay. Zoras Lee Grant has questions. Let's have a look. Could you please put your question in the Q&A box? I can tell. Well, that's quite a lot. Okay. So the first question is TGF-β or senescence caused by lack of loop exclusions by cohesion?
No, we don't think it is a direct effect on the loop or the formation of the loop. I cannot say too much. We have now we have an hypothesis with what could be the link between any PBL and indeed the TGF-β cell senescence pathway. But it's not a direct effect of TGF-β on the loop.
Right. Okay. The second question is from Catherine. Any in fetus diagnostic or treatments could be possible, you think?
In fact, yes. It could be possible, but as you know, it could be possible because there is more and more patients which are indeed detected during echography, at least when people are aware about this syndrome. But unfortunately, this is detected when the syndrome is very severe. So where people can see some really cardiac defect or see even some problem during bone. But yeah, you could think that you can treat, but as you know, it's very, very difficult to do pediatric trials. So that's why we are now more focusing on a treatment after birth, specifically for the neuronal defect.
Right. Great. The third question is that, actually there are two questions for these ones. What is the mechanisms by which the, sorry, it's not, yeah, what is the mechanism by which the MIPBL genes mutations leads to the CdLS disease? Yes, yeah. And the second part is, does the TGF-β inhibitors and treatments of other cell type inhibit senescence?
So the first question is, I showed you that NEPBL is a loader of the coenzyme complex onto DNA. So if you have less NEPBL, you can expect that you will have some problems to load the coenzyme complex on DNA, and specifically when it is required for the loop of the enhancer on promoters. That's why there is a lot of problems in gene transcriptions. So this is mostly the problem of this syndrome. There is a lot of gene transcription defect. So many genes that should be expressed are not expressed, or they are not expressed at least at the right time during development.
Yeah. Right. So the fourth question is, actually, I have this question as well. Basically, I think she's asking why you need have you looked at another like markers for senescence? Is that my question? Also, I have a question on this as well. Is the programmed embryonic stem cell senescence really different from so much different from adult cell senescence? Can we use like beta glycosidase staining kind of acids to test the senescence on the programmed embryonic stem cell senescence?
Yeah, this is a good question. Yes, indeed, there is a beta galactosidase positive cells during embryonic senescence. We use P21 because, for example, P16, which is a lot used in adult senescence, is never expressed in embryonic senescence. So there are clearly differences. There are some common features, but there are differences between embryonic and adult senescence.
Right, great. Finally, I have one more question for you. So we know that actually the TGF-β signaling is involved in many cellular pathways, like controlling cell proliferations and survival, as well as embryonic stem cell differentiations. Do you think you just use the have you looked at the proteomics and RNA sequencing analysis like you did with the mutants on with the TGF-β inhibitor treated cells to look if any like changes with the gene expressions?
No, it's a good question. We have not checked that yet. But what we know is that by using the inhibitors, using in fact any kind of inhibitors, when you use a drug, you never block at 100% the pathway. So we think that we are basically, we are hoping that we are blocking the over activation of the pathway, but not the pathway by itself. That is a basic pathway.
Right. So you think actually the dosage of the inhibitor will matter? You're actually not like turning it off, but you're actually tuning the signaling down in those mutations.
That's
okay.
correct. Yeah,
okay.
exactly. And that's why when you look at the clinical trial for patients, cancer patients, there is no side effect of this drug.
Yeah, we have another two questions. And how can we differentiate between cellular senescence and the cellular persistence when dealing with cancer therapies?
I'm not sure I understand what does it mean, cellular persistence? Do you mean persistence or quiescence?
Yes, yeah, I think maybe it's the difference between the programmed embryonic stem cell senescence and the cancers, maybe it's cancer cell senescence.
Cancer cell senescence. This is a good question. We are looking a lot. We are from the literature, because we did a lot of didn't show you here, but we did a lot of single cell sequencing for another project about programmed cells, embryonic cell senescence. And we compare indeed with some markers that had been found in cancer cell senescence. Again, there are clearly some common features, but we cannot say that this is exactly the same process. But I would speculate it could be, because we know that during development, a lot of things which is normal during development is just dysregulated in cancer.
Okay. Right. Yeah, we have one more question from Michael's. What do you think is the contribution of a cardiac neuron crest was first or second half field to the cardiac phenotype in CdLS?
Yeah, it's a good question again. In fact, we look very specifically at that, and we don't think that there is a problem in the neural crest cells, even the migrations. So there is, I would say there are 2 points I would like to highlight. What I showed you about septations, this is a septations which is due to the migration of second heart field progenitors, not the neural crest. The neural crest participate much later in the aortic development. And another, there is one, in fact, an English lab published years ago where they deleted any PBL in the neural crest, and there was no phenotype at all. At least no cardiac phenotype.
Okay, I think that's it. Thank you very much, Dr. Michel Puciet, for such a fascinating talk today. Thank you all the audience for coming, attending this webinars. There will be more exciting MC webinars coming in the middle of June. Next time, the topics will be on tissue-specific immunologies and immunotherapies. Once again, Medican Express is committed to provide a platform that enhances scientific communications and accelerates scientific discovery. So please stay in tune. And I'm looking forward to seeing you all very soon. Goodbye now.
Thank you. Bye-bye.
Thank you. Bye.
Bye.